I can introduce the next speaker, Cristiano Garrino from the German Federal Institute for Risk Assessment. He has gained expertise, especially in food allergy, allergens, and contaminants. Currently works on insects as novel foods and new sources of allergens. So please, Cristiano, the floor is yours.
Welcome, everyone, and thank you for inviting me. So today I will introduce you briefly to the concept of food allergy. Then I will present you a case of an approach to allergenicity risk assessment. Then I will speak about the recent EFSA scientific opinion on allergenicity assessment of food and feeds derived from biotechnology. And lastly, I will present you an example of a risk assessment applied to insects as food. So food allergy is described as an adverse reaction to an otherwise harmless food that involves an abnormal response of the body's immune system to a specific proteins in food. The prevalence of food allergy within the general population is quite high, and symptoms can vary from very mild to severe anaphylactic shock and eventually death. It is an obvious social and economical impact since people with this type of condition rarely consume food outside their household. And it's important to study it also because there are causes that are still unclear. And even though immunotherapy has made big progresses, there is not a general cure for it. There can be different immunological mechanisms involved. However, the most common is the one mediated by immunoglobulins E. In this type of pathway, we recognize usually two steps. The first one is called a sensitization phase, where the immune system is firstly exposed to an allergen and specific IgE are produced. And then there is a second step called elicitation, where following the second exposure to an immune system, the allergen is recognized and the chain of events that leads to the onset of the symptoms is activated. Different novel foods have already been recognized as potential elicitors of food allergy, either because they are able to sensitize or because they are able to cross-react with known allergens. Not necessarily the ingredients present in novel foods are newly introduced in our diet. Sometimes these are old ingredients processed using new technologies that might have an impact on their allergenicity. And of course, it's worth to mention that also new trends can introduce novel allergens in our diet. So I would like to clarify the difference between primary sensitization versus cross-reactivity, because this is important for what we shall say afterwards. So in a classical example of primary sensitization, the person first comes into contact with an allergenic food, for example, an apple. And then following the second exposure to the same allergen, he starts developing the symptoms. On the other hand, in the example of cross-reactivity, the patient is sensitized by something else, for example, in this case, pollen, through sometimes a different route of exposure, in this case, inhalation. And then after coming into contact with a food allergen that mimics the allergens contained in the pollen, can develop a reaction. This is, for example, the classical pathway of sensitization of apple allergy in Central North Europe. We now move to see how allergenicity is assessed in novel foods. In 2021, EFSA updated its guidance on how to write an application for a novel food. The default assumption is that because novel foods contain protein, they do have an allergenic potential. And because there is not a single test able to certify the ability of a certain food to provoke an allergic reaction, the approach remains the one based on the so-called weight of evidence. This approach foresees to focus on some characteristic of the proteins, such as their stability, their resistance to digestion, and their degree of homology with known allergens. This weight of evidence approach is the same that is applied to evaluate the allergenicity of novel proteins in GMOs, and it was initially suggested by a document of the Codex Alimentarius in 2003. However, this approach, which was thought for a single protein newly expressed in a GMO plant, for example, presents different flaws when it's applied to a complex food containing many proteins. Moreover, what this approach does not assess is the capacity of the new protein to de novo sensitize non-allergic individuals in the population. This strategy was challenged in the course of this cost action called Imparas, which ran from 2014 to 2018. 3 years ago, the authors issued a review to summarize what they have learned during the course of the action. Basically, what they aimed to do was to go from an approach based on studying the amino acid sequence and the resistance to pepsin of the protein to a more comprehensive approach evaluation, which could also take into account, for example, the history of the novel food, as well as to include the sensitization potency of the novel food within the risk assessment. The work was divided into different working groups. Group number 1 re-evaluated procedures for allergenic risk assessment of GMOs for their application to novel foods. The authors summarized their findings in a review paper published in 2018, where they identified some gaps and action to be taken. They stressed the importance of considering the impact of food processing techniques and food metrics. And in general, they highlighted the lack of reference materials and standards. Most importantly, the working group discussed the use of protein pairs. This is a combination of an allergenic and a known or a weak allergenic protein in order to develop potential comparators for future assessment. Working group number 2 focused instead on in vitro methods to predict de novo sensitization. Their work, led by the Netherlands Organization for Applied Scientific Research, allowed the publication in 2017 of a first adverse outcome pathway for food allergic sensitization. Within this pathway, there are 5 key events and 3 molecular initiating events. This is the original figure from the publication, which is a little bit confusing. This is a clearer picture published this year. The first key event is represented by the epithelial activation, where the allergen goes through the gut barrier. In the second key event, the antigen is uptaken by the antigen-presenting cells. The third key event is the migration of the dendritic cells into the lymph node. In the fourth key event, antigen-presenting cells interact with the T lymphocytes and prime the T helper 2. And finally, in the event number 5, there is a B cell isotype switch that leads to the production and release of specific IgEs. We skip working group 3 and go directly to working group 4 that focus on risk assessment and dissemination. The group had several tasks, one of which was to define possible risk management targets and their implication of future methods development. The idea is that the risk assessment strategy should be developed following the inputs of the risk manager in order to respond to its needs and goals. So the questions should be, what risk do we want to prevent? For example, if the risk manager goal is to prevent sensitization, then only novel foods that do not have any sensitizing potential will be accepted. This basically would mean that every novel food would need to be safer than any known food. And this is extremely conservative and probably would lead accepting no novel products. At the other extreme, we could decide to accept all the novel foods that present a low risk of anaphylactic shock, in which case, as you can imagine, practically all novel foods would be accepted. So this is an overview of possible parameters and criteria for risk management decision-making. This include hazard-based, exposure-based, and risk-based approaches, and they can be applied both to the sensitization and to the elicitation phase. So the Imparas cost action main conclusions were that a network of expertise is needed to support integrated risk assessment strategies, and there is also a need for a clear outline of a decision-making criteria for risk managers. There is also the need of consensus of an assessment strategy for the risk of de novo sensitization. In vitro methods should be focused on the developed adverse outcome pathway for allergy sensitization. All methods should be harmonized and validated using specified reference proteins and extracts. Protein pairs should be used to rank existing known allergic proteins according to their allergenic potency. Finally, the knowledge of how different matrices and food processing impact impact on allergenicity should be investigated more in depth. Now I will show you how these concepts were implemented into the new EFSA scientific opinion on allergenicity and protein safety assessment of food and feeds derived from biotechnology, which used to be the basis for the drafting of the previous guidance on the preparation and submission of an application for authorization of a novel food. So the opinion was published last year and it was drafted by the EFSA panel of GMOs. The main aims were to define knowledge gaps, identify specific research needs, determine how the current methodology can be improved, and prioritize research funding. Some highlights for the summary. The weight of evidence approach remains valid, and the allergenicity safety objectives need to be redefined by addressing key questions for risk assessors and risk managers, like what is the purpose of the risk assessment, what should be assessed, what level of confidence is necessary, and what are the unacceptable risks. The authors introduced the concept of clinical relevance. An allergen becomes clinically relevant when it causes symptoms, and this clinical relevance should be the key driver for the development of the risk assessment strategy. Sensitization is a predisposing risk factor, but it's unrelated to clinical relevance. Criteria could be the severity, the potency, the prevalence, the exposure route, and the level of exposure. The authors also point out that a set of non or low allergenic proteins is needed. And this is the concept of the protein pairs that was proposed by the IMPARAS network. The reason why some proteins become allergenic in some individuals is not yet fully understood. Although generally food allergens are organized into protein subfamilies, there are no single common structural causes that can contribute to their overall allergenicity. Very recently, a theory was developed according to which there are biologically active small molecules that could play a role in the sensitization process in addition to the allergenic protein itself. And this was nicely presented during the last IACI Congress in June by Professor Heimo Breiteneder. Then the authors start presenting a series of tools for the prediction of allergenicity, starting from the in silico tools. These are generally algorithms that rely on existing databases and try to study the homology of an unknown protein to known allergens. So if a relevant shared sequence identity is observed, then subsequent IgE binding studies will follow. And these tests will use sera of allergic individuals who are reactive towards the homologous allergen. So these in silico tools only focus on the cross-reactivity of potential, and there is no in silico tool at the moment that provide information on the capacity of the proteins for the de novo sensitization. There are several publications of these algorithms. This is just one example of a recent paper from the same group that was involved in the IMPARAS cost action. An important topic is the evaluation of protein digestibility. So processing, digestion, and transport should be evaluated. However, several studies demonstrated there is a poor correlation between resistance to pepsin digestion and allergenicity. So the evidence that supports the resistance to the degradation by pepsin as a direct predictor of allergy is weak. Alternative in vitro digestion methods that use physiological conditions might be more useful. One example is the InfoGest protocol, which was elaborated in the course of another cost action and is currently the internationally recognized method to evaluate static in vitro digestion of proteins. This protocol has now been used in several works, and this is an example of a peanut digestome, where the authors focused on a peanut allergen that was originally thought to be non-resistant to digestion, while by applying the InfoGest protocol, the authors identified resistant IgE binding peptides. More in general, the measurement of protein digestibility should not be regarded as a standalone endpoint. Assays that evaluate the IgE binding are considered adequate. The sera involved in these assays should have a convincing clinical history, and the elicitation of allergic symptoms should be established by a double-blind placebo-controlled food challenge. At the same time, sera from individuals with other kind of allergies should be used as a negative controls to exclude non-specific IgE binding. The authors of the opinion also proposed the idea of building a bank of monoclonal specific IgE, which could be used in the future to detect allergenic proteins in order to overcome the issue of collecting sera from patients. Another important in vitro prediction tool is the basophil activation test. This was consistently proven to be highly specific and highly sensitive for the prediction of the clinical outcome, and it could be used as to dispense patients from undergoing oral food challenges. As for the in vivo models, at the moment what we have is not predictive for allergenicity. Therefore, it is impossible to use them for the risk assessment because there are no standardized predictive models. Also, it will be ideal to anyhow avoid the use of animals. Finally, I'm going to talk about insects as novel food allergens. This is a very recent story published on a local Italian newspaper of a person allergic to crustaceans who ate a piece of focaccia prepared by using crickets, flour, and had an allergic reaction. This is a typical example of an allergic reaction induced by cross-reactivity. So currently, 6 insect-based products have been authorized in the European Union. 2 of them are based on the yellow mealworm, 1 on the migratory locust, 2 on the house cricket, Acheta domesticus, and 1 on the lesser mealworm, Alphitobius diaperinus. In 2015, EFSA published the risk profile related to the consumption of insects as food. And regarding the risk of allergenicity, the committee said that this is plausible and may be based on the existence of common allergens of arthropods and other animals like mollusks and nematodes. The close phylogenetic relationship between the different arthropods may explain sequence homologies and be responsible for possible cross-reactions between edible insects and other arthropods. So according to this opinion, the risk is mainly associated to cross-reactivity. So we have the evidence that insects can cause symptoms in individuals previously sensitized, for example, to crustaceans due to the cross-reactivity of their allergens. But are they also able to primarily sensitize new individuals? The scientific evidence of it was provided in 2017 by Brockman and colleagues, who analyzed a small group of mealworms farmers who were frequently coming into contact with mealworms due to their work and did not present any sensitization or clinical symptoms towards shrimps. These farmers underwent a double-blind placebo-controlled food challenge with mealworm snacks, and 2 out of 4 subjects confirmed their reported history of food allergic symptoms. This was a scientific proof that mealworms are able to sensitize new individuals, most likely via skin contact. This is an overview of the 6 scientific opinions issued by EFSA on the 6 approved insect-based products. There is also a 7th opinion on the safety of UV-treated mealworm, which has not been yet authorized. I took an image of one of the abstracts, but the text is actually quite the same in all of the opinions. And what the authors say is that this novel food may induce primary sensitization and might cause allergic reactions in subjects with allergies to crustaceans and dust mites. However, the panel concludes that the novel food is safe under the proposed use, which means that the sole scientifically proven possibility of this novel food of inducing an allergic reaction was not considered enough to deny the authorization. Now we see how the risk manager implemented these opinions into the EU legislation. So this is the current the regulation of 2023 authorizing the placing on the market of Alphitobius diaperinus. And you see in comma 2 of Article 1, it says the annex to Regulation 2017-2470, which is the Union list of novel foods, is amended in accordance to the annex of this regulation. In the column of the annex describing the additional labeling requirements, it is marked that the labeling of these foodstuffs shall bear a statement that this ingredient may cause allergic reactions to consumer with known allergens to crustaceans and dust mites. The statement shall appear in the close proximity of the list of ingredients. Finally, I will show you an example of a new approach to allergenicity assessment of insect proteins carried out by the group led by Anna Sancho at the DTU of Copenhagen. This is actually the report of a former EFSA fellow who spent his fellowship in this group 2 years ago. The approach is described into 5 main steps: literature search, protein preparation, a series of laboratory analyses including the protein characterization, the analysis of digestibility, and the use of rats to perform in vivo analysis, the in silico analysis, and a last step of literature search, research and report writing. The EUFORA technical report only displays the methodology and does not show the results, which were, however, presented in the course of the last IACI meeting by Anna Sancho. I took a picture of the poster so I can show you the main highlights. The poster was on the allergenicity assessment of the black soldier fly, and it's focused on the results of the in vivo tests. So basically, a group of rats were immunized with different doses of black soldier fly, as well as with 2 preparate controls of high and low allergenic compounds, namely peanut and spinach. The results showed that in terms of immunogenicity, the black soldier fly stands something in between peanut and spinach. And in terms of the sensitizing capacities, is closer to spinach than to peanut. So the conclusions were that the black soldier fly proteins have a low level of allergenicity compared to that of peanut, and ingestion may pose a low risk of inducing allergic reactions in shrimps allergic individuals. Thank you for your attention.